Alrighty, welcome back everyone. We are going to be starting chapter 3, and that's going to be a discussion of the biological macromolecules. We have 4 main groups that we're going to be talking about: the carbohydrates, the lipids, protein, and finally nucleic acids. All right, so before we get started on the individual macromolecule groups, I want to talk about a very, very important concept, and that is the way that macromolecular reactions occur. Basically, the macromolecules that we're going to be discussing have monomers, which are their small molecules that they're made up of, the units that they're made up of. That's the smallest unit that they can break down into. And we have the polymers. And the polymers are when you take the multiple monomers and link them together. The reactions that we're going to be talking about are how you take these monomers, these individual units, and combine them to form this large polymer. And then you go in the opposite direction, taking these polymers and then breaking them down into monomers. So when we take individual monomers and link them together, we are undergoing the chemical reaction known as dehydration synthesis, or a dehydration reaction. The dehydration reaction will then result in the polymerization of the molecules. So basically you're taking the individual monomer here, the individual monomer here, you're linking them together, so you're forming this bond. And the reason it's called a dehydration reaction is because you are losing a water molecule in order for the bond to form. So dehydration reaction, you are just basically creating a bond, and it is usually with the hydrogen of the one monomer, the hydroxyl group of another monomer, that will result in the formation and eventual loss of the water molecule. And then that resulting loss of the water molecule creates this covalent bond. And depending on what type of macromolecules we're talking about, the specialized covalent bonds that are formed will have names. So if you think about it, you can keep on adding monomers to a chain, and you can have a polymer that's going to be thousands upon thousands upon thousands of monomers long. So that is the dehydration reaction. A very, very important concept in Bio 181, because you're going to see our discussion in Unit 2 when we talk about the different reactions that are occurring in living organisms. The opposite type of reaction is a hydrolysis reaction, and that's where you're taking the polymer and you're breaking it down. Hydrolysis, from the word hydro, which is water, lysis, to split or break. So basically you're performing a reaction that is using water to break the bond. So what you're doing is essentially you're adding water into the reaction, and the monomer that is getting broken down is going to result in the addition of the hydroxyl group and the hydrogen group to the monomer. And can continue to do this, continue to do this, continue to do this, continue to do this, all the way down till you get individual monomers. So I want to go back to dehydration synthesis for a little bit. So you can think about this as taking one amino acid and another amino acid, combining them together to form a polypeptide, a polypeptide, a polypeptide, and then you have this huge polypeptide that'll eventually form a protein. With the hydrolysis reaction, when we consume, for example, proteins, you'll take that large piece of protein, whether it's in the form of fish or steak or beef, and you'll take that large piece of protein and break it down into the individual monomers, which are the amino acids. And then you take those amino acids that we consumed that came from the foods and you rebuild them. Okay, so that is the introduction to the way that macromolecules undergo chemical reactions. So we're going to start talking about carbon, because you're going to find out that the backbone for all the molecules, or the macromolecule that we're going to be discussing, is carbon. Carbon is highly unstable because it wants to share its electrons. It wants to share the electrons. So when we talk about organic molecules are usually molecules that have carbon as their backbone. Okay, so organic equals carbon. Carbon usually will combine with hydrogen, so you have what's known as hydrocarbons. And most of the time you have the addition of oxygen, and then you can also have the addition of nitrogen. So we know that carbon has 4 electrons in its outermost shell, so it wants to form bonds, and it will form different macromolecules that you are well aware of. For example, the gas carbon dioxide, proteins, and sugars. All right, so if we take these carbons and add hydrogens to them, we are going to have hydrocarbons. And you can see that the bond angle is a 90-degree angle. That means there's equal sharing of the electrons. If there's equal sharing of the electron, we're going to describe that molecule as being non-polar. In order to have macromolecules or molecules react, we must have some sort of functional group that is going to allow reactivity. The different types of functional groups that you can have are the hydroxyl group, which is the OH, the carbonyl group, which creates the aldehyde groups, the carboxyl group, which creates the carboxylic acid, the amino group, which creates the amine, phosphate group, which creates the phosphate molecule, and then you have the addition of phosphate molecules, for example, adenosine 1,2,3 triphosphate, otherwise known as ATP, and finally methyl groups, which are very, very important for the nucleotides when we talk about DNA and RNA. All right, so we're ready to move on to our first group of macromolecules that we are going to discuss, and those are the carbohydrates, the carbs. So you probably heard the word monosaccharide and disaccharides right here. What are saccharides? Sugars. So mono means one. So that's going to be quick sugar, or quick, energy from the simple sugar, the monosaccharide, and then the polysaccharides, which have two main functions in biological organisms, storage, and also they can help with the structure of some of the biological organisms that we'll be talking about. You can see the carbohydrates in multiple types of carbohydrate, breads, pastas, grains, so on and so forth, that will result in the complex carbohydrates. But then when you look at the individual monomer, it is glucose. And what happens is glucose is basically linked together via dehydration synthesis to create these large polysaccharides. And the thing about the polysaccharides is that they are able to store energy because the bonds within the molecules or between the individual molecules will store energy. And the energy type is potential energy. So a really important note that we have to make here is carbohydrates are the primary fuel source for cellular mechanisms. So important that we have an entire chapter dedicated to the breakdown of sugars in the process known as cellular respiration. So again, you can put these definitions on note cards. Monosaccharide, mono meaning one, saccharide sugar, single sugar, polysaccharides many. So you can have multiple sugars linked together, multiple glucose molecules linked together. So there's the monosaccharides, simple sugars, simplest carbs. When broken down, they don't make more carbs. So you're breaking them down into individual, just atoms, basically. They're easy to digest. They're a good and quick energy source. So you get the energy immediately because it's very easy to break down. So our three main monosaccharides that we talk about in biology: galactose, glucose, and fructose. Galactose, glucose, and fructose. All right, so glucose. Glucose is the most important carbohydrate for living organisms, okay? Glucose is the blood sugar. For example, that's the sugar that is floating around right now in our bloodstream and being taken into our tissues, okay? And the way that we regulate blood sugar is via hormones that are produced by the pancreas. One that you've heard of that breaks down glucose, insulin, and another that builds complex carbohydrates, which is glucagon, which actually produces glycogen. Okay, so let's take a look at this diagram right here. So you have glucose, which is blood sugar. You can use that for energy, and then you break down the glucose through a process of cellular respiration to produce ATP. When you don't need the energy, you can store and convert the glucose molecules by linking them together to form glycogen, which is then stored in your liver and in your muscles. And then third, long term, you can take those glucose molecules, link them together in a reaction and form, yes, you guessed it, the dreaded fat, okay? So fat is a good energy storage molecule, and the reason that you gain fat in your body is because you've consumed a lot of glucose and you gotta figure out a way to store it. Okay, so as I stated, glucose is important because of the process known as cellular respiration, which we're going to talk about. And basically it's taking the glucose molecule and breaking it down into energy in the cell. And we're going to talk about that process in great detail. And the energy that is released, that is the ATP that is released and produced, is used for all the processes in a cell. What you require for cellular respiration to occur in most organisms is oxygen. So you use these general equation for cellular respiration right here. Glucose, which is C6H12O6, with the addition of oxygen, will yield water, carbon dioxide, and ATP. That is the energy. So remember, ATP is the energy currency in a cell. Complex carbohydrates, the polysaccharide, it's more than one sugar building block. Okay, so I mean, you take an individual monomer, for example, glucose, and you combine them together and you can form the polymers. Now, before you can actually use the energy from the carbohydrates, you have to break down that large chain. So it takes a little bit longer to break it down, and it's a slower process. So basically, that's why we typically will use carbohydrates as long-term energy, because you have a quote unquote time release fuel source. So you'll know a bit prior to like a big football game. I remember we used to have to consume carbohydrates, pastas, because you could take those and it could be broken down over a period of several hours and you could have continuous energy. There are also indigestible structural components that we're going to talk about in the complex carbs. All right, so we have some disaccharides that are pretty important that I'm sure you've heard of, at least two of the three that are listed here. Lactose, where you have one glucose and one galactose coming together. So lactose is milk sugar. Sucrose, which is glucose and fructose coming together, so that forms table sugar. And then maltose, which is malt sugar, which is basically two glucose molecules coming together. What type of reaction is occurring to take this glucose and the galactose molecule to form the lactose? Yes, a dehydration reaction or a dehydration synthesis reaction must occur. And it is the same to produce sucrose and to produce maltose. So what type of reaction is going to occur when we take sucrose, table sugar, for example, the sugar that you consume in, let's say you added sugar to your coffee, okay, this morning. What reaction is going to be required in order to utilize that sugar? Yes, the hydrolysis reaction, where you add the water to break the bond. Very, very important, okay, to understand those concepts, the concepts of breaking down and building macromolecules. Some common polysaccharides that I know you've heard of, okay, starch. Starch is a polymer of glucose. You find it as a storage sugar in plants. It's, for example, you see in bread, flour, pasta, cereals, grains, and potatoes. Cellulose, which is a glucose polymer that's found in the cell wall of plants, and it is fiber. It's not digestible. We as humans do not have the enzymes to break down cellulose. And finally, glycogen. Glycogen is the storage form of glucose in animals. So we find it in us, in our muscles, and in our liver. All of these polysaccharides are structural isomers. Okay, so complex carbs, you can get quick energy release, eat an orange, or you can get time released energy, for example, eating a bowl of oatmeal. Okay, so you can see a graph here, time versus blood sugar level. You have the initial spike, you get the sugar going into your bloodstream, you use that energy, quick bursts of energy, and then here looking at this table, or I'm sorry, this graph, you have a long drawn out release of the energy. Really cool there. All right, again, monosaccharide, polysaccharides, the carb. So that's really cool image right here, right? Croissant, bagels, some pasta, breadsticks, bagels again, and donut, okay, to form carbs. Yeah. All right. So that is discussion of carbs. Next group we're going to talk about are lipids, and lipids are very, very different because they have multiple structures in terms of the way that they look, but they have one common feature, and that is they are non-polar, or they are hydrophobic. Yes, hydrophobic, okay? So features that you see here, non-polar, remember they don't dissolve in water, greasy to the touch, and a significant source of energy storage. You can see 3 different types of lipids, the fats, the sterols, which are going to form steroids and form cholesterol, which help regulate growth and development, and then the amazing phospholipid molecule, which comprises the membranes on planet Earth. All right, fats are a type of lipid, so long-term energy, okay? All fats have similar structure, okay? For fats, they have a glycerol head, so that means that it is polar, but then they have the fatty acid tails, which are chains of carbon hydrogen, which make them non-polar, okay? So dietary fats, you have triglycerides, 3 tails, 1, 2, 3. So glycerol, which is a type of alcohol, plus 3 fatty acids produces the triglyceride. The energy stored in the carbon-hydrogen bond is a great source of energy. So remember that you still have carbon, you still have hydrogen, and you have oxygen. So that's why you have the hydrocarbons and you have the oxygen. The solid at room temperature, that's what RT stands for, solid at room temperature are the fats, and the liquid at room temperature are oils. So you can see a fat cat here, very fat cat. That's a happy cat when you see a fat cat, right? All right. So ester bonds link the hydrophilic and hydrophobic portion of the triglycerides. So the glycerol, you remove 1, 2, 3 hydrogen from here, 1, 2, 3 hydroxides. So you're losing how many water molecules to form a triglyceride? Try 3. 3 water molecules. So there it is, your loss of the water molecules, the dehydration reaction forms the triglyceride, and the bond that connects them is an ester bond. You can see a difference between saturated and unsaturated fats. You've heard these terms before, I'm sure. Okay, saturated means that you have a carbon-to-carbon single bond. Okay, and the saturated fats are solid at room temperature. Butter, margarine, shortening, so on and so forth. unsaturated fats, excuse me, are where you have carbon to carbon double bonds or even triple bonds. And again, they will be liquid at room temperature. So the unsaturated or polyunsaturated fats are the healthy fats. Also the fats you find in nuts or legumes, almonds, walnuts, peanuts, cashews, whatever it is that you like to eat. Those are unsaturated fats. And the unsaturated fats actually can be used to break down the saturated fats. So this is something that they stated in the mid-90s. Basically use fat to break down fat. That's the easiest way to actually break down the saturated fat or the fat that you find in the human body. All right. Next, we're going to talk about the second group of lipids, which are the sterols. For example, this is cholesterol, and you can see, again, it's going to have the characteristic of being non-polar, okay, non-polar. So cholesterol is the base structure or macromolecule that's responsible for making the steroid hormones. And again, if you have a large buildup of cholesterol or you consume a lot of cholesterol, that's where you have bad cholesterol. Organisms need cholesterol. Organisms produce or synthesize cholesterol because cholesterol is very important in the cell membrane, especially in animals, because the cholesterol molecule allows the cell membrane to be fluid. So it gives fluidity to the cell membrane. The other important thing with cholesterol is the formation of the steroid hormone. So you can see the function of the steroid hormone regulating the sexual development, maturation, and then the sex cell production. Estrogen influences memory and mood, and testosterone stimulates muscle growth. Yes, men and women, excuse me, let me go back, have both estrogen and testosterone, but as you know, women will have more estrogen than testosterone, and men will have more testosterone than estrogen. But if you look at these molecules, you can see they're very similar. The only difference is the functional groups. This is a hydroxyl group here, carbonyl group here. Okay, and then you add here, you have a methyl group and then another carbonyl. Here you just have a hydroxyl group and then a hydroxyl group here. So you can see the difference between the estrogen and testosterone is basically a difference in the placement and location of the functional group. Rather fascinating. The last group of lipids that we're going to talk about are the phospholipids. And the phospholipid are responsible for the creation of the phospholipid bilayer, which I talked about earlier when we talked about water. Okay, remember it has a phosphate group, which is polar, a fatty acid group, which is non-polar. So you have a hydrophilic head and you have a hydrophobic tail. Very, very important. So lipids, characteristics. First and foremost, you should know all lipids are hydrophobic. And then you have the different types. Triglycerides, which are long-term energy storage. The sterols, which are basically used for structural and regulatory functions. The phospholipids, which make up the cell membrane. And then finally, the waxes, which are used as protective coating. Very, very important with the lipids. So again, that's a second macromolecule group that we are talking about. All right, so I'm going to stop here and I will continue and pick up our discussion with protein in a little bit.